This lecture on genome size and organization is going to be divided into two to try to keep the individual video lectures a little bit shorter. So when we start talking about size, genome size, organism complexity is loosely correlated with genome size. That means, in general, the more complex the organism, the bigger the genome is, the more genetic material there is. Genomes tend to get larger over time rather than smaller. That is to say that they accumulate genetic information, but they generally don't lose it. Sometimes it happens, but in general, genomes just accumulate more information. It's sort of like that closet in your house that you never clean out. You just keep putting more stuff in there even if you don't use it. And some of it you go in and take out, but the rest of it just stays there. You don't get rid of it. But it's not really hurting anything. It's just sort of hanging out. The size and the complexity of a genome varies just as much as the organisms on our planet. In the first example here, we have a bacterium, Helicobacter pylori. It's a single-celled organism, a prokaryote, with a very relatively small genome. Then as another example, we have Saccharomyces cerevisiae. This is yeast. This is another single-celled organism, but it's eukaryotic, so it has a larger genome. And then increasingly more complex, we move on to animals with a larger genome. Humans, more complex than a fly, even larger genome. And then plants in general tend to have very large genomes as well and may not necessarily be more complex. So this Norway spruce and this whisk fern, many people would probably argue is less complex than a human, but has a really huge genome. So genome size is loosely correlated with biological complexity, although there are examples in both directions. But in general, the more complex, the larger an organism is, the more genetic material they can contain. Now, the genome contains information from the evolutionary history of that organism and the current function and phenotypes of that organism. So it's a record as much as it is an instruction manual. So you can look at changes over time. You can trace changes in a genome over time, and often those changes are related to the complexity of the organism. So genomes increase as complexity of the organism increase, but genomes rarely decrease. If a gene in a particular organism is no longer needed or no longer provides an advantage, it may fall into disuse. But they aren't just dropped out of the genome. They stay in the genome, and they're just not maintained in the same way that a gene that is being used regularly is. So again, this is like your closet where you throw stuff in and some stuff just accumulates in the back. You might not use it anymore, and if you think you're going to use it, you might find that it is broken. In general, genes are not recycled or reused, although they could be. Sometimes genes are sort of repurposed, their sequences are sort of repurposed. Genes can acquire mutations to make them dysfunctional or to help them acquire new functions. Mutations generally occur at random, and if that mutation renders the gene dysfunctional, it's not going to be used in that organism anymore, and that could have detrimental side effects. Maybe not. We'll talk about that a little bit later. But when it becomes dysfunctional, there's no more maintenance. So this means that changes will continue to accumulate, and there's nothing to stop those changes because that gene is not being used and there's no positive selection pressure anymore. Another possibility is that you might acquire a new function. So you can think of this as sort of recycling or repurposing some of those unused things in your closet. Maybe someday you need something and you're like, oh, I could use that. It's not perfect, but I could use that. The same thing sort of happens with your genes. If you have multiple copies of similar genes, those other copies can acquire new functions within that organism based on evolutionary pressures. So I want you guys to think about how this process of not reducing a genome is actually biologically beneficial, and also what effects you might have if you actually start removing pieces of the genome that you don't need. So I want you to think about this a little bit. I will also set up a discussion board for this and just sort of think about or discuss this idea of acquiring or accumulating genetic information, and then also why you wouldn't want to reduce a genome size. So think about how environmental changes, and new selection pressures arise. And how some genes may suddenly become useful. And then also think about what would happen if you were randomly deleting, or even not necessarily randomly, but just deleting old material. What might the negative side effect of that be? So your book uses this sweater analogy, and it's similar to the closet analogy I was just talking about. The book says that your genome is like sweaters in your closet. You buy new ones when you need new sweaters, when old ones are worn out or get too small or whatever, or you just change your mind about what you like, you acquire new ones. But if you don't throw them away, this is sort of how the genome works. And of course, you might come back to one later and decide you like it again. And this is sort of how a genome might repurpose genes. So think about those things, and just think about this question, and discuss how these changes or lack of change in size of a genome might affect the evolutionary history of an organism. All right, so let's say a gene actually loses a function. So all of your cells have all of the genetic information in your genome, but they don't use all of the genes all the time. And of course, environments change. So at one point in time, you might have a gene that's particularly useful, and then the environment changes. Maybe that gene is either detrimental or just no longer needed. If genetic material is not under some sort of selective pressure, meaning that different varieties or different variations of the genes provide some sort of fitness advantage, if that's not happening, those genes can actually accumulate inactivating mutations. So let's look at this cave fish example. If you're a cave fish, a fish that lives underwater in a cave with no light, you don't really need your eyes. If you're a fish that lives on the surface where there actually is sunlight, you do need your eyes. So there is a selective advantage to keeping that gene with the correct sequence. So if a fish on the surface develops a mutation with no eye, they're going to be selected against. That variation is going to be lost. But what happens if an individual in a cave loses its eye function? Well, really nothing. It wasn't using it to begin with. So not having an eye is actually not detrimental. So it might actually be more beneficial to not expend energy to maintain all of those processes that are related to the eye. And so those processes, those genes that are related to the eye might actually fall into disuse and not be maintained. Now, of course, loss of an eye means that cave fish can't ever leave the cave because they'd be in an extreme selective advantage. That means they're going to be confined to the particular environment that they're in. And if they're not going to leave anyway, that's okay. So sometimes, though, genome reductions may occur, and sometimes pieces of DNA that's non-essential can be lost from genomes, and this is usually related to basically the metabolic cost of maintenance. So it's actually more beneficial to the organism to lose the genetic material that it isn't using than it is to maintain it. Overall, though, that doesn't occur. If a gene loses function, the sequence of DNA remains within the organism and it just sort of hangs out. It just doesn't have a purpose at that particular time. So let's move on from genome size to genome organization. Bacteria are our prokaryotes. And one of the characteristics of prokaryotic genomes is that they contain generally one chromosome, and that chromosome is a closed circular piece of DNA. So the scribble here is actually a large circular piece of DNA, and that contains most of the genetic information for the organism. It contains all of the required information for that organism to live and to grow. Bacteria also have plasmid DNA. So plasmid DNA is a small piece of sort of expendable DNA, and this plasmid is going to be basically something that confers a particular environmental advantage. So this could be something like antibiotic resistance, or a plasmid might encode the ability to utilize a particular food source. Now the key here is that they're expendable. They give the organism some sort of advantage, but is not necessarily required for them to survive. Now the other thing that's cool about the plasmid is that plasmids can be shared. And so this is one way that bacteria can exchange genetic information. They don't reproduce sexually like prokaryotes do, like most prokaryotes, and so they have to have other ways to exchange genetic information. A lot of the knowledge that we have of the molecular processes, such as DNA replication or transcription, translation, things like that, are best studied in bacteria, and this is because they are simpler organisms. They're easier to work with. But most of those processes are often very similar in eukaryotes, just slightly more complex. So moving on to our eukaryotes, or generally multicellular organisms, eukaryotes tend to have more complex genomes. They are often made up of multiple chromosomes, and they are always linear. So eukaryotic DNA is packaged into a chromosome structure. Now the chromosome structure is made up of our helix of DNA. That helix is wrapped around special proteins called nucleosomes, and those proteins help to basically compact and fold the DNA so that it can fit into our cell. This complex of proteins and DNA is called chromatin. So in order to get all of the genetic information that we have in our cells to fit, we have to sort of pack it in really tightly, and we do this using special proteins. Before we get to that, let's talk about the overall structure of the eukaryotic chromosome first. So I'm going to talk about this in more detail in just a minute. One side of this X is actually a single chromosome. So this is one chromosome, and this other side is an identical copy. So just keep that in mind. I'll come back to this a little bit more in a few minutes. So let's talk about the physical layout of this chromosome. The end or the tip of the chromosome is called the telomere. Now, the telomere is really important for sort of protecting the ends of the chromosome. The center, or almost the waistline, is called the centromere. So this is a highly compact region of DNA, and it's really important. We will talk about the centromere much more extensively when we get to cell division, because it's important in actually helping to line up the chromosomes and separate them correctly. Each of these guys sticking off is an arm. By convention, we name the short arm the P arm for petite, meaning short. And this is important when we start talking about mapping, when we talk about locations on chromosomes, because we'll say a particular gene is on the P arm or the short arm. The longer arm is called the Q arm. Nothing fancy about that. It's just because Q comes after P. So P for the shorter arm of the chromosome, Q for the longer arm of the chromosome. So the human genome is made up of 23 pairs of chromosomes. And we can see those chromosomes in this picture that we call a karyotype. So a karyotype is basically a computer image of the chromosomes in a particular cell. Now, these chromosomes are always metaphase chromosomes. You might remember that word from your general biology class. Metaphase is one of the phases of mitosis, which is cell division. And in metaphase, the chromosomes are very tightly compact. Now, each of our chromosomes has a specific structure, sort of an identifying structure. And the way this is made is by taking a cell in metaphase, staining the chromosomes, and then, not these colors, but staining them so you can see them, and then digitally enhancing them. So these ones have been enhanced by color so that we can tell the difference. But you'll notice that chromosome 1 has 2 long arms. It's the largest chromosome. So in a human karyotype, chromosomes are numbered by size. So chromosome 1 is the longest one, and it contains a very specific set of genes. Now, we have 23 pairs of chromosomes, or 23 copies of our chromosomes. So we have 23 types. You can see those here, 1 through 22, and then our sex chromosomes, X and Y. This one shows just 2 X chromosomes. So this is going to be a female because she's XX. A male would be XY. And the Y chromosome, let me get green here. If we had a Y chromosome, it's actually just a real small chromosome. So here we have 2 X chromosomes instead of an X or a Y. So before we move on, you get your pairs from mom and dad. So each of your parents is going to, in fertilization, give you 1 copy of each chromosome. So mom and dad, and then you have 2 copies of each type. So the human genome is made up of 23 pairs, or 46 total chromosomes. So this picture shows the special type of staining that can be done to a human chromosome. And you'll notice, again, we're numbered largest to smallest. Chromosome 1 is always the largest, 22 is the smallest, and then we have our sex chromosomes. In this case, we actually have a sex chromosome here. So all of these chromosomes would be lined up and analyzed via computer. That's how we get our karyotype. But in a cell, you'll notice they're just sort of a jumbled mess. So in a cell, chromosome 1, which is, I don't know, maybe this guy, maybe that guy, are 2 different copies, not really sure. Chromosome 1 is not necessarily associated with the other copy of chromosome 1. So in a cell, they don't really associate too much. But these bands help us to distinguish another characteristic of our chromosome. These are one of those characteristics is called heterochromatin. So heterochromatin are the densely stained areas. So we can see that here, all of these dark stains. Heterochromatin is highly compact DNA. And that DNA is usually information that is not expressed as much in an organism. Heterochromatin often has a physical function, such as being the centromere. So we can see some heterochromatin there. And it might just be regions of DNA that aren't used very often, and so they can be more tightly packed. Then we also have euchromatin, which is the less densely packed DNA. So that's the lighter staining here. We've got some light staining, light staining. Those regions are going to be more often expressed. So this is DNA that is used more often. So it's less compact so that the expression machinery can get in there and work on it. All right, so let's talk about this chromosome packing that allows a lot of genetic information to be folded and compacted down into our cells. So I really like this picture here. This is sort of a cartoon picture that shows the packing, and then we have an electron microscope image, which is a special type of microscope that can see really small details. So we have our chromosome here. This is our DNA molecule, and it is wrapped around those nucleosomes. And they are wrapped around nucleosomes in a regular pattern that allows the DNA to be compacted. So each nucleosome has 146 base pairs wrapped around it. Now, the reason I say that is just to show you how precise this packing is. And then there is a regular amount of DNA in between the packing. And then we also have some other proteins called histones that aid in the packing. So nucleosomes and histones sort of wrap around the DNA, wrap the DNA up and tighten it and fold it up. So the histones and the nucleosomes are positively charged, and this is because DNA is negatively charged. Remember that those phosphate backbones are negatively charged. So this allows them to interact with histones and nucleosomes. So that facilitates the packing. When the DNA needs to be accessed for replication or expression, the DNA will be sort of unraveled from around the nucleosomes and opened up. This is an image that's not in our book, but I like how it demonstrates how this folding actually happens. Now, I'm not worried about you knowing all of these very specific details, but I do want to point out what is actually going on. So after the nucleosomes and histones wrap up your DNA, it actually sort of coils and folds back on itself, producing what we call a 30 nanometer fiber. So nm equals nanometer. It's a really tiny measurement. Nanometer, that's the width of the fiber. Now, what is actually happening here is you can sort of visualize if you have a string or a shoelace, something like that. If you hold either end of your shoelace, hold it at either end, hold one end still and twist the other end, and just keep twisting it, you'll see this pattern. It begins to wrap around itself and fold up tightly. And eventually you get this really regular looped pattern, and as it continues to condense even more and more, that 30 nanometer fiber gets a little bit thicker because it's folded in on itself, and then that one folds even tighter until we eventually get that metaphase chromosome. And this is the chromosome that we see on our karyotypes. Now, this packing is important because each of our human cells has about 2 meters of DNA. So if we stretched out a single stretched out all of our chromosomes and laid them end to end, be 2 meters. That's about 6 feet. And each of our cells contains all of that information that is compacted into the nucleus of the cell. So that is how important all of this folding and wrapping and twisting is for our genome.